I'm gonna wait one more minute. To remind students. Okay, so I'm gonna go ahead and get started. I'm gonna start with a brief introduction. Okay, so good morning, everyone. I'm currently in Atlanta at the American Chemical Society conference. I actually have to head out, so I apologize to our guest lecturer that I won't be able to attend to the whole lecture. But I want to give you an overview of her background. So she obtained her bachelor's in the Faculty of Chemistry Biology at the University of Santiago of Chile. Went on to obtain her PhD in chemistry, also at the University of Santiago of Chile. Her doctoral thesis was titled Obtaining Gold Nanospheres and Gold Nanorods for Potential Bioapplications. Her supervisor was Dr. Marcelo J. Kogan. Since then, she has held a number of positions. She was first a research support specialist, high throughput and spectroscopy resource center at the Rockefeller University in New York City. Then, in the same location, became the manager of spectroscopy. She then moved on to be a staff scientist in the chemical biology and therapeutic department at St. Jude Children's Research Hospital. Went on to then in the same St. Jude's research operations manager and is currently the director of scientific research. She has a number of publications, over 15 publications. And so with that introduction, I would like to turn over to Carolina.
Thank you, Arthur, for this kind introduction. So I'm gonna share my screen. Oh no, wait. Um, can you see the presentation?
I see your desktop.
Oh, okay, sorry. I'm not very used to Zoom. We use WebEx here.
Yeah, I'm sorry about that. Yeah.
No, that's fine. Uh, this one. Can you see it now?
Yep. Yes,
All right. And do you see, wait, is that the right?
that's correct. Yep.
Perfect. And then, okay, so thank you for the introduction. My name is Carolina Adura. I'm the Director of Scientific Research here at the Chemical Biology and Therapeutics Department. And today I'm going to talk to you about the CBT centers. And in conjunction, all these centers the main goal is to bring chemistry to medicine. So we're trying to develop new potential therapeutics and also discover of chemical probes that can be used to understand diseases and pathways. So even though Arthur already gave like a very kind introduction, I want to start just telling you a little bit about my journey. So I'm from Santiago, Chile, and I received my PhD in chemistry from the University of Santiago. And then I spent half of my training at the University of Barcelona in the Institute of Research in Biomedicine. So I then moved to New York and I worked 7 years, as Arthur mentioned, as a research specialist and then as a manager of spectroscopy of the High Throughput Screening Research Center. And here I was in charge of developing new high throughput screening, developed biomolecular interaction assays, in instruments like isothermal titration calorimetry, surface plasmon resonance, etc. And I was also in charge of train people and maintain the two NMRs that the institution had at that time. After that, I joined the Chemical Biology and Therapeutics Department in 2020, and I started as a staff scientist in the Cupido lab. I think he gave a talk to this group a couple of weeks ago. Um, So I helped him started his lab when we moved from New York to Memphis. Then I moved to the position of the manager of research operations, where I oversaw all the shear instrumentation in the department. I work in the capital equipment inventory, which is we have to account for more than 500 capital equipment, meaning at the time of the purchase, it was a certain threshold, and those are considered assets of the institution, so we need to account for those. Um, so then when I was the manager of research operations, I learned about the centers in our department, and I tried to understand how they were working on and how we can develop a pipeline that is a drug discovery pipeline here at St. Jude. So I worked with the chair of our department, Asim Ansari, and we started developing that pipeline. So then I was promoted to director of research, scientific research, and this is what I'm doing currently here. This is my current role, and the most important part here is that I try to bring all the centers together and pull resources from the different centers, so then we can engage in different collaborations with faculty in our institution, in our department, and also faculty outside of St. Jude when the time allows and permit that we can collaborate with those. So just to give you brief overview, what I'm talking about when I talk about the collaborative centers. So as I said, these centers, and I'm gonna talk about all of them in a minute, and I'm gonna show you a few videos. So these centers integrate they form an integrated pipeline, and the purpose is, as I mentioned before, the discovery of chemical probes and drug candidates here at St. Jude. So the pipeline is structured sequentially like operations that will start with a hypothesis that a lab will bring to us. Then we will set up aims and milestones, and of course we will set up and try to be very realistic on what the outcomes can be from that. The capabilities that we have here mirror leading biotech pharma operations, and they're rarely accessible for academic institutions, even here in the US. So we're very lucky. We are subsidized by the institution. So we try to use most of the resources that we have in a very fairly and efficient way. And I'm going to talk about that process later in my presentation. Um, how do we work with the how we help the chair to allocate the resources that we have. Now, here at the institution, there are shared resources that are fee for service, but by design, we are a collaborative department, so we don't charge for the work that we do. If you have heard about hydrop screening, maybe in other institutions here in the US and even in other places in the world, there's a small fee because it's very expensive to run high-throughput screening of small molecules, and I'm going to go into the process also for those that don't know. But it's basically testing thousands of molecules in a particular target until you find something that has the response that you're looking for, and then you work with that molecule and you try to optimize it to see how far you can get. So it's very important to mention that we're 100% subsidized by the institution, so we don't charge for any work that we do. And then importantly, these collaborative projects between our department and different faculty or different researchers here at St. Jude are a very close partnership, meaning we provide the expertise in chemical biology, but we do need the expertise of biologists here at the institution. So when projects come to us, we're always trying to look who is going to be the champion of that lab. So who is going to pursue the molecules that we're going to find, for example, through this high throughput screening, or the molecules that we're going to be optimizing, and how those can be tested, hopefully up to animal models. So that's the whole goal of the centers. So to provide you a little bit of understanding of the biology of using small molecules. Our department, of course, focuses on the use of the small molecules to study biology. So you need to think in small molecules as precision tools that can be used to modulate the function of biomolecules in different cell lines. For example, proteins, right? We're looking for small molecules that we can use to control the function, activate, or inhibit, etc. So small molecules are also very important to understand the structure of these biomolecules and what is the relationship between the structure and the function. So the small molecules are a very powerful method for dissecting also signaling pathways. So for example, in a disease, a protein has a mutation, and then that changes the conformation of that protein that make the protein not work anymore, and then that will generate, for example, a disease. So in that case, what you want to do, maybe you want to find a small molecule that will help you restore the function of that mutated protein, so then you can address that disease. Making a small molecule to put in the organism of a person is very complex, and it takes years and is super expensive. But what we do here is we generate the knowledge, the base knowledge for that, and we try to discover the starting material. And of course, later on down the road, we will have to engage with industry partners if we really want to put this forward and put it in the clinic. So just to give you an idea here, in 2024, there was a total of 50 FDA-approved new drugs. Out of those, more than 50% were small molecules. And why small molecules? Small molecules are cheaper to produce than biologics, and by biologics, I mean maybe a CAR T cell, a CAR T treatment that includes you have to retrieve the cells from the patient, maybe treat them, then put them back. That is very expensive. Probably you have also heard about CRISPR screening or modifying the structure of the DNA in order to fix the disease. That is also very expensive. Small molecules is not. And so far, most of the treatments that we have in the market are small molecules, ibuprofen, acetaminophen, etc. Those are small molecules similar to the structure that I'm showing here in my slide. And of course, are more accessible to patients. So once the patents are filed and they're licensed and then they can be produced, it's more easy for patients to get them. So if you imagine, like lately, there's been some advance in sickle cell disease treatment where you can do a genetical modification for those patients. The countries that get sickle cell disease do not have these capabilities, and you need to put the patient in the clinic, you need to make the treatment. So that's very complex. I'm not dismissing it. I think it's a very powerful tool and is where the future, as where all these drugs are going into the future. But today, they're not accessible for many of these patients. So small molecules are. You transform it in a pill that you can distribute and people can have it. But the big challenge that we have here is small molecules are not easy to be specific for a target. And that means if I design a small molecule and I put it in a cell, it might hit not only that protein that I wanted to fix the structure, it might hit other 2 or 3 proteins that are working normally, and that will generate a side effect. So this is one of the major challenges that we have in the field, is how we make small molecules that are biologically compatible and they're also specific. So to overcome this challenge, CBT, the Chemical Biology and Therapeutics Department, has constructed a compound collection that is a small molecule library collection over the years. Okay, so our department has just turned 20 years. Last year in 2025, it was funded in 2005. So over all this year, we have been adding small molecules into our collection. So we have around 600,000 unique molecules. We have those molecules repeated. So we have several copies of those. In total, we have around 4 million to store in a big building with a big freezer that is all automated, that you can retrieve the molecules, etc. So we have different type of molecules. We have all the FDA approved drugs that have been over the years. We update this collection every couple or 3 years. So we include every molecule that have been already approved. And then we have clinical candidates as well that were molecules that tried to get to the FDA, but for a reason they failed. Now, what do we have this? Because here clinicians like to use these molecules for repurposing. And what that means, cancer, catastrophic diseases in children are not very valuable for pharmaceutical companies. So there's no many small molecules designed to treat catastrophic diseases in kids. So if a molecule has been approved for a disease in human, that means it passed all the safety controls. And then what physicians do here, they take those molecules and they test them to see if they can be repurposed and if they can be used to treat children's diseases. So this is what we call repurposing. So it will start with patient samples. Here at St. Jude, we are lucky to have directly access to patient samples. So they will test these molecules and they will see, for example, if they specifically kill a particular cell line that is involved in a particular tumor of a childhood disease or a childhood cancer. And then they will say, oh, this works. It's going to be way easier to move that FDA-approved drug into a clinical trial with kids than if we start from zero. Now, this is not an approach that works in all the cases, so this is why we have other type of molecules that is basically starting from zero. So we're going to start with a molecule that has never been tested, and then we're going to try to move this forward. Another type of libraries that we have are the target-based libraries. For example, there's many diseases that involves kinases proteins, that involve GPCR proteins. So over the years, researchers have been studying these different proteins, and they have developed small molecules that not necessarily are in FDA approved, etc. But what if I want to understand how a particular kinase is involved in a particular disease? Maybe I can use a kinase inhibitors library and I can see how that modify the pathway, etc. So that's why we have these target-based libraries. Now we also have a large part of our library that is what we call drug-like, meaning it's a small molecule that looks nice, it doesn't look toxic, and then we buy thousands to companies. They just make these libraries and they make 100,000 molecules and they sell it to us. And they don't have a particular function, but we test them with our particular targets or disease model, and we see if they have any effect and a positive effect that we're looking for. And then that molecule might be modified to be a clinical candidate, hopefully, or just to be a chemical probe that can be used to understand diseases, as I have been mentioned before. So what we can do with our collection, we can do repurposing screening, we can find novel chemical matter, we can also work in drug combination, that we have a few labs here that work on that. So is there any synergy between two different molecules that will potentiate when they're together and that will improve treatment, etc. And we also do virtual screening. So we know the structure of all the molecules that we have in our library. So people will dock every single of these molecules in the structure of a particular protein and see what will bind to that protein and maybe generate the effect that we're looking for. We have another library that is very popular that is called the Mechanism of Action library, that we use for target and pathway discovery, to linking known targets with new biology. So these molecules, this library is called Mechanism of Action because we more or less know what these molecules do. For example, this molecule we know is targeting a particular protein here, for example, that is involved in this pathway cascade. So by adding this molecule, I might stop this and I can see what's going on. When I block the pathway here, maybe I have another molecule that blocks the pathway here, and then that's going to generate a different effect on the mechanism that I'm looking for. Now, we like to see this as what we call genetic loss of function, but this is transient. If you do a CRISPR screen, and I'm not sure how familiar you are with that, but if you change the DNA and avoid that cell produces that particular protein, that is going to be definitive. So you cannot going back. But with small molecule, you can, because the small molecule will enter the cell, will do its job. If you don't replenish with that small molecule, the small molecule will eventually be degraded and will be eliminated from the organism. And sometimes this is what researchers are looking for, just to generate a transient blocking of the pathway to be able to study what comes next. So how does this work in our department? So our centers that are 7, and we are just working on hiring the leader for an 8th center that hopefully is gonna come soon. We're trying to develop and leverage the chemical tools to address therapeutic challenges in catastrophic pediatric diseases. This is the focus of our centers. So everything starts with a hypothesis. So a partnering lab will come and will say, look, I think this particular protein or this particular cell line has this particular phenotype, and I would like to understand through chemistry how this is happening, or can I find something that blocks the function of this particular protein? So they generate an hypothesis and they come to us and say, I want to test my hypothesis with small molecules. So then in general, this is going to be a disease-linked gene or a dysfunctional cellular pathway. So then at the end here, we're going to have a chemistry-based tool. So they're going to come with an idea. They're going to work with our centers. They're going to come with an idea here, hypothesis, work with the centers, and we're going to have an output. So what is the input first? Different project types. So it can be advanced or exploratory, somebody that has already known that this particular protein, they have what we call is target validation. They have done several biological experiments to say, this is the protein that is linked to the disease. Let's just go here, let's enter the pipeline, let's test molecule, let's improve the molecules, and let's see what comes out of this. We can also have exploratory projects. Somebody that said, I might think this is the protein that is involved in this. Can we do some testing? So we also accept those type of projects. We also build collaborations, and then that can get to connect people from different departments and different expertises. For example, we are working on a big project with sickle cell disease. We have one of the world leaders of sickle cell, and he came to us, and we are generating this partnership, this collaboration, and we are working with them. And finally, we do technology development as well, where our centers will work in developing different new technologies that will help our partners that come with a hypothesis to try to prove it. Now, when all of these enter this pipeline and this circle that you're seeing in the middle, that all the centers are collaborating together, then we need an output. And what are examples of outputs that we have had? We have had, for example, the SynTef, that is what Jose works in the Ansari lab. Those are molecules that bind to the DNA and can generate changes in transcription of different genes. Also, we have other type of molecules that also will generate an impact in transcription. We have molecules for new targets. We have an anti-malaria molecule that I'm gonna talk about this in one of my slides about the story that is in clinical trials. We also have this that is a coenzyme A activator, so it increases the production of coenzyme A, and this is also in clinical trials. We have also developed some antibiotics here in the Lee lab, that is one of the laboratories in our department. Um, in terms of technology developed, we have helped labs create libraries of building blocks in thousands of those. Building blocks are the small molecules that you're going to put together to generate a bigger molecule. We have also worked in degradation. I don't know how familiar you are with protacs and with molecular glues. Those are molecules that hijack the proteasome, meaning they're going to bind to a protein, bring it together, and bring it to the proteasome for degradation. So instead of blocking the function of a protein, you're just getting rid of the protein that's being produced in the organism. So now I'm going to give you a few examples of these compounds that have come out of the research that we do in our lab. So, sorry. Malaria is one of those. This project started in 2010 and is still in clinical trials. So we're talking about 15 years since the molecule was discovered. So malaria is a disease that affects almost 300 million cases in the world, and most of these are children under 5. So what happened with this molecule? In 2000, right, so in in 2010, we published this, I wasn't working here, but this paper came out of our department where they screened 300,000 small molecules against the malaria, so the parasite that generates malaria. So then they did some SAR, meaning structure activity relationship, where this molecule, the chemists work in changing different parts of the molecule, the core, and then these rings, etc. And they came with developing a first clinical candidate. So they were 3 years where they took the original hit from the screening, they work around it, and then they generate this molecule that is called the SJ733. So and that was published in 2014. So then in 2016, we had the first clinical trial that started as a phase 1A. Then in 2020, the results of the phase 1B was published in the Lancet Infectious Diseases journal in 2020. And then finally in 2021, this molecule entered phase 2 and is now there in clinical trials. So this is an example of what we can do. We have done very little about this because it's very hard to go from the beginning to the end, but it is possible. And this is what we're trying to do here in our department, to advance something from the research lab into the clinic, into clinical trials. Another molecule that I mentioned in my previous slides is the PANK3 activator. So pantothenate kinase associated neurodegeneration is a disease that arises of the mutation of one gene called PANK2, and that leads to coenzyme A deficiency. So what happened? In 2015, they took this protein that can be purified in the lab and they did biochemical assays. They tested more than 500,000 compounds, and they looked for activators and for inhibitors of this protein at the same time in the same assay. Everything was developed here at CBT. So then they came with this molecule that has an EC50 of around 900 nanomolar. And then again, the chemists work on this and improve the molecule, making different modifications, and they make this molecule 800 times more potent. So then what happened with this? They tested in different conditions, and then they take the CLOP, the oral viability of the molecule, what is the half-time of the molecule in plasma. They started, of course, with mice, etc. So this molecule was really good. And then to do all this improvement, they work with a collaborator in structural biology department that his name is Stephen White. He's retired now, but it was very key to have the structure of the protein the structure of the molecule and being able to dock this. So that will shed some light on what modification needed to be done in the molecule in order to bind better to the protein. Finally, this molecule that you see here was sold to a company, and this company did a few modifications, as you can see here. And this molecule entered later in clinical trials and is still there. So we'll see what happens. So this is to treat patients with acidemia. This is an activator. Contrary to the previous case that I showed, it was an inhibitor. So it inhibits a protein that allows the parasite to propagate. Instead, here we are activating the function of pancake. So coenzyme A can be produced in the body, and then you can avoid a disease that is called propionic acidemia. Now, this program is still going on, and the PI, his name is Richard Lee, that started this program, is now looking for inhibitors of this protein because it has been demonstrated that inhibiting this protein can be beneficial to treat some cancers too. So there's a lot of work to do here, and he has this big program that he's working with several researchers here at St. Jude. Now, that brings me to my last example, that is degradation. So molecular glues are small molecules that bind on one side to your molecule, your protein of interest, and on the other side, they bind to a E3 ligase. So the E3 ligase, what they do is they ubiquitinate the protein that is close by to them, and that ubiquitination leads this protein to be attracted to the proteasome, and the proteasome will degrade the protein. So one of these E3 ligase that is very famous is called cereblon, and it's been used for the past 15, 20 years. And there's a few molecules now in clinical trials with this new mechanism. So what did we did with a GSPT degrader? So our department built a library of more than 5,000 compounds that have the capability to bind to cereblon, and then hope that on the other side of the molecule that you can see here can bind to a protein of interest and bring together cereblon, that is this E3 ligase, with your protein of interest. Now, this library is unique to CBT and St. Jude. It's more than 5,000 compounds. Right now, it's more than 6,000 compounds, and it was funded by the Alex Lemonade Stand. It's a foundation for childhood cancer, and it was funded, of course, by the institution to be able to generate all this. So what they did is they took different cell lines of different modalities, and they test the whole library, expecting to see death in one of those cell lines. And if you have never heard about cytokine screening, you have different molecules in a 384 well plate, where each well has a different molecule, and then you put your cell line or your protein on top of it, and then you look at each single effect in each single molecule. So they found this molecule that was active, and what they did is they mutate the cell line, and they knock out cereblon, meaning what happened if I don't have cereblon? And what happened is there was no effect. So it's this curve on top here. So that means the mechanism for which these cell lines were dying is because cereblon was attracting another protein into the proteasome for degradation. So this protein, so what they did is, after testing and making sure the mechanism is through cereblon, they did some proteomics, meaning you add this molecule to your cell line and then you do mass spec on it, and you want to see which proteins were maybe overexpressed or downregulated by the effect of this molecule. And what happened here is that this protein, the GSPT family, it was the one was completely degraded. And then after doing some research, they discovered that this GSPT is a family of protein that are involved in translational regulation. So they're highly expressed in pediatric hematologic malignancy, like ALL, AML, etc. So blocking or degrading this protein is beneficial to treat this type of diseases. So after they had the molecule, they know the mechanism of action that is through cereblon, and they know in which protein this molecule is degrading. They test this molecule in different in vivo models. So here they develop this molecule. They tested for bioavailability, and it was highly orally bioavailable, which is pretty good. Some of these tests are done, most of these tests are done, of course, in mice that have like with the disease model. But sometimes the molecules work better when they are injected rather than given orally. So you want to give a medicine, a drug orally to in the future to patients. Yeah, you don't want to be injecting stuff all the time. So these high oral bioavailability, and then at this lower concentration that they were testing it, the mice tumor was reduced. So it was very highly efficacious. There was a high efficacy in vivo for the PDXs that are the models that the mice that contain the disease, right? This is the model. Finally, this molecule, we licensed it. We didn't have the capabilities here at St. Jude to keep developing. So basically, we look for companies that were interested on this, and they literally buy the patent for $2 million. And now the patent is theirs, and they are working on it, hopefully to develop something. So these are like three different examples of what I was talking at the beginning, how small molecules can be used. So they can be used as activators, inhibitors, or even degraders. So basically in our portfolio, this is how this will happen. So this is the curves of the project. So you're going to have assay development that is being done mostly in the lab. Then you're going to test thousands of molecules. You're going to identify a few molecules. You're going to do structural relationship activity. So you're going to be doing some changes and understand what parts of the molecules are the ones that are important for engaging that target. Then you're going to do some lead optimization that is going to be preparing these molecules and make them better, make them more bioavailable, less toxic, etc. And of course, finally, we would like to test them in vivo. So some of the portfolio that we currently have are these type of molecules. So we have been developing kinase inhibitors or degraders all the way to test them in vivo. Ligands for different E3 ligases. The body has different E3 ligases like that cereblon that will help to bring proteins for degradation in the proteasome. So the most common ones, as I mentioned, is cereblon and another one that is called VHL, but there's several ligases, that are around 600 ligases. So can we leverage that, and can we find ligands for those ligases, and can we use those ligases to degrade other type of molecules, maybe tissue specific, a ligase that only get expressed in the skin, and then you can treat skin cancer, etc. And so on. We have different other projects. So in terms of the repurposing that I was talking before, there's a different clinical trials that might not started here at CBT as the ones that I show you, but we have been part of it, and we have been helping clinicians here in the institution to come to develop these clinical trials. So now I'm gonna talk a little bit about how we collaborate. I think we still have like 20 minutes. So we have these 7 centers, and the idea is we start with an initial contact, and this is where I play sort of a key role, because people will come to me and they will express their willingness to work with us, but they also will tell me what type of disease they're working, what is that they want to do, where are in the stage of the project, if it's exploratory, if it's more advanced, what do they need? Do they need us to provide some compounds only? Do they need to engage in the whole pipeline? Maybe they only need to do some chemistry. So then I try to set clear expectations for them of what we can do and what we cannot. And then they will move into sometimes brainstorming sessions when they will come and we will be together with faculty in our department and with the center leaders. And then we will listen to them, we will listen to the story, we will decide if it's something that we can handle and how we can help them. So then they're going to start in general with an exploratory phase. So they will be paired with one of the centers just to do proof of concept assays and see if we can miniaturize the assay so we can test thousands of molecules, or if the chemistry of the molecule that they already have can be optimized if it's a difficult chemistry, or if there's already patents there and it's not worthy to try to work on that, etc. And then we will go to an optimization phase, and sometimes we call it automation. If it's going to work with all our robotic system, we have several robotic systems that I'm going to show you in a minute. And then can we validate the assays? Can we test? Can we do some initial testing of small molecules, etc.? When we see the project is robust, it meets all the requirements that we have to engage in a scientific project, then we ask them to present to a committee that is called TRAC. So TRAC is the Therapeutic Discovery Resource Allocation Committee. So basically we need, as I mentioned before, we don't charge for doing any of this, but we need to allocate the resources that the institution provide in a fair way. So this will work as the person that wants to collaborate with us. Once they have a robust assay developed, they will present a letter of intent, and then this will be evaluated by the committee. We will get together, we will read the letter, and then they will present to the committee, where we are going to evaluate the significance of the proposal, the investigator expertise. Maybe somebody that is a computational biology wants to do some in vivo testing, and we will say, well, you might need to engage with a biologist that does this. So this is what is important to verify the investigator expertise. What is the innovation? Are we gonna compete with big pharma that are doing the same? Maybe it's not worthy, we're way smaller. And as I mentioned at the beginning, if there is a champion, a partner from that lab that is gonna help us to do all the biology later on. So then milestones will be established, and then resources will be allocated. And by resources, I mean mostly people that work in the different centers. They're going to be working with in this particular project, and this is the resources that we will allocate. And then after a year, we will review. Sometimes some projects will end up there and will finish in publications, in grant applications, or in patents. Or sometimes the project is going so well, but it still needs another more year to be more solidified and to be able to publish and to apply for a grant. So then we will review where the project has been advanced so far, and then we will decide if we will provide resources for another year, and then they will come back to the committee again. So this is how more or less we try to handle our portfolio. And then this committee is integrated by members from our department here, the chemical biology, but also different other departments. So this is sort of an unbiased way to evaluate different projects. So this is one of the centers that I was mentioning before. This is the Compound Management Center. This is Mariana. She's the manager of this group. So as you can see, we have different robotic system automations here. These robotics move all along. And what they basically do is they formatted our small molecules. So this is the big freezer that I was talking before. That contains 4 million tubes. So this machine will move, we will program the computer and we'll say, I want to test 3,000 molecules, and this little picker will go and just pick up those 3,000 molecules, and then they will be bring to this lab, and using this other machinery, those molecules are going to be placed in different plates, and then the researcher can screen them or test them in their biological assays. So basically, they maintain all the 600,000 molecules. They deliver them, they purchase new molecules, and then we store them. In the case of Jose, his lab works with a particular molecules that have been developed in his lab, so we store them here, and whenever they need it, they just submit a request, and this team will provide the molecules to them. We then have this other team that is called the High Throughput Biosciences, that is led by Tao-Shen Chen. He is a member of our department, and he's also a director of this center. And they're going to be doing all the testing. So people, collaborators will come with a particular cell line, and then this team will be in charge of putting the molecules that were requested to the compound management team and test them in different cell lines and see which molecule works better. So they work in target validation, so making sure we are targeting what is causing the disease. They will do assay development, miniaturization. As you can see, everything works in these tiny, tiny wells in 384 well plates. So when people work in their lab, maybe they're working in test tubes or in 96 well plates. We want to do it in a high throughput fashion. So we want to test thousands of molecules at the same time. That's the role of this center. They also run some high content screening, meaning instead of looking for a luminescent signal in a plate reader, they will look at the cell and they will take pictures of each well that contains cell lines, for example, and they have a fluorescent agent that will turn on when the cell is dying, just to put an example. We also have a siRNA screen, meaning we can target particular proteins and they can be blocked for translation. So this is why you use this RNA screen. So that can be used in combination with small molecules, for example. We have 3 chemistry centers, and they are divided by the medicinal chemistry team, targeted protein degradation, and analytical technologies. So what happened is the medicinal chemists are the ones doing the changes in the molecules. They will, if you have an original molecule and they need to make some chemistry around to make it more soluble, more cell compatible, less toxic, this is the team that is going to work on that, the medicinal chemistry. And then the Analytical Technology Center is going to be the team that is going to test those molecules. They're going to do some ADME profile, biophysical properties of the molecule, making sure the molecule is soluble, is stable, etc. They also produce all the preclinical formulations for the whole institution. So by preclinical, I mean putting different drugs in mice, and those need to be prepared in a particular formulation that is not going to be toxic for the animal. So all of this is prepared here by the Analytical Technology Center. And finally, we have the targeted protein degradation. That is the team that developed that molecular glue library. And they also design products that are molecules that are 2 different molecules that have a linker in the middle. And one molecule binds to your protein of interest and the other one binds to the E3 ligases, and they will put a linker to put those 2 together, and then hijack again the system of the proteasome. Those are products. They're different from the molecular glues because the molecular glue is a smaller molecule that binds, that generates a proximity just by binding to your cereblon, that is your E3 ligase, and maybe producing a conformational change on that molecule, that in that protein, that will allow another protein to bind to it. That's a molecular glue. A product is a larger molecule that we know from one side bind to your protein of interest and from the other side will bind to the E3 ligase, and they work with different linkers. So all part of this optimization is done here in this center. We then have the lead discovery informatics team. led by Anand, and then they take care of all the cheminformatics of the department. You can imagine we generate tons of data. So all this data needs to be analyzed, needs to be stored. So they work on that. They also do all the analysis of that synergy that I was mentioning when you try to test more than one molecule together and see if there is any synergistic effect. You also need computation to be able to analyze those data. Then they do molecular dynamics. They can do structure-based design, so they can have the structure of the protein and then the molecule that binds here, and then computationally they can imagine different molecules that can bind to the protein structure, and then they can work with the chemist to see if they can do it like the real molecule. They also do molecular modeling and design and virtual screening, meaning they take all the proteins and all the molecules that we have, the structure, and test them in different structures of proteins just to see if anything binds to them. And finally, we have project management office. As you can imagine, with these large teams that we put together, we need a project manager that help us with complex projects where there are multiple PIs. We need to sign agreements of confidentiality, material transfer when we are working with different institutions. We need to do some data sharing. We need to schedule meetings. We need to take the minutes for those meetings. All of that is part of what the project management managers do in our department. And with this, I'm done with my presentation. Thank you so much for listening, and I'm happy to answer any questions that you might have.
Okay, I want to thank Carolina again for, for such an informative lecture. Sadly, Arthur had to step out due to travel restrictions, so I'll be kind of leading the remainder of the discussion session. So yeah, if anybody has any question, please unmute yourself and feel free to ask Carolina.
And you can ask any question if it's related to my career path or what I do here or etc. Any question that you might have, I'm happy to answer.
Okay, so it seems like people are being shy. I have a question at least. I mean, I know a lot of what you do from, he's just working around with you, but it just as a point of right, if you come to the centers and as a person who just kind of wants to start up a project, is it also available, or do you have to already come out with a full-fledged project when you decide to meet with any of the cores or the relevant centers to what your project is supposed to be about?
No, not at all. We accept, the communication is always open. So we talk to anyone that is planning, even in the future, to do something, or they just have an idea. They're welcome to come talk to me. Sometimes they come and talk to me, and then we meet with some of the center leaders as well. If I believe it's going to be a better fit for them to be talking to these people, etc. So absolutely no need to have a full project to come. Now, if they want to engage in larger projects and we know that's going to take tons of resources from our centers, then is when we have them presenting to this committee. But informal conversations happen all the time. And if there's something small that can be finished in a week or so, we will be happy to help everyone.
And I guess I'll continue asking questions. When it comes to, when you have these compounds that you might be interested in taking to clinical trials in some cases, um, does the centers and the cores, I know you manage a good chunk of them. Do you guys also take care of all the patenting work, all the or at least putting the labs in contact with the relevant legal A's to start the patenting process, as well as to take them schedule or work with potential clinical trial like procedures?
Right. And we just had a seminar this morning with the Office of Development and Patenting. And there's a whole team here at St. Jude that will help with that. But of course, if it's a small molecule that was made here and it was synthesized here, but by our chemists, they will help the whole process. And then, as Asim was mentioning this morning, you want to make sure who are the contributors to that patent, who own authorship on that, and then everything is fine. We file tons of patents every year, and maybe not all of them make it to a licensing. And licensing means somebody else will come and pay you money to get that patent information, and now they are the owners of that. But we need to protect everything that we made here because potentially that could be used in the future. So yes, we file tons of patents a year, and everything is done here at CBT with the help of this technology licensing department.
Anybody have a question? No, doesn't look like it. So I have one final, just broad. Like you said, you mentioned the labs, right? Like these are a bunch of different fields, a bunch of different labs with different specialties that, comprise all the cores and all the centers at the CBT and in general, and when it comes to drug discovery. And people only think about, one lab did this and that's what's present in all these papers. Can you maybe like, kind of talk a little bit about the hardships or the difficulties of having to, work in these like collaborative environments, but at the same time, the advantages of doing these type of collaborative work where one lab handles one chunk and then passes it on to another, and then everybody kind of works together at the end.
Right. So that's a very important question. And I will say the advantages are giant, right? Because you can have the different expertise from different labs. So our centers are expert in miniaturized assays, test the molecules, understand if the molecules are generating or not an effect that you are desiring on a particular system. Now we have the expertise of the chemist, so they can see the structures, they can see if the structure are viable or not, if they're going to be toxic, if there's a particular group that we need to remove, etc. So there is a huge advantage on having different expertises and mostly to have the biology. So in that examples that I show you, for example, the one of the product, the molecular glue that is the degrader, of course, it was a huge team. It wasn't only the chemists. There was chemists, biochemists, biologists that they were dosing the mice, that then the chemists that analyzed the blood of the mice to see if the compound was degraded or not. There was people from the proteomics facility that helped us to understand the mass spec data to arrive to the target, what was the target that was being degraded, etc. So challenges are one team work faster than the other, and sometimes that happened to us. We are very fast at screening molecules, and if the partnering lab that is doing the biology, if they don't have a champion that can test the molecules in a different cell line, for example, or even if we have a molecule that we analyze the properties and it's good to go and it's good to be injected in the mice. If they don't have the model, if they're just working on the model, we already generate all this work. We already did everything that we could. But then we're stuck waiting for them. So that's one of big challenge. The other challenge is it can be some duplication of efforts. So this is why it's so important to have a project manager that handles all of that and that everybody is honest and tell that project manager what they are doing, what problems are they facing, and then that person can oversee the whole process. So when projects are too big, I'm also included on those. So I can even see from a vision even further from the project manager, and I can see what everybody's doing, what are the needs, where the project is getting stalled, and then what resources we need to inject on that. So collaboration is key, and honesty, meaning we want to hear from everyone. We want to have regular meetings with everybody present the data and everybody present the challenges that they have. So then those can be overcome. And having someone like me that can help to identify also what is missing and where we can get it from. Maybe we need a computational biologist in the team. So I can reach out to the computational biology department and see if somebody is interested in joining the team, etc.
Yeah, I think that does a good job of highlighting, how the differences between just normal academic research and more drug discovery research, right, in chemical biology, where in academic research you kind of just have a lab, a few grad students and a postdoc working on it versus, more a more broad setting where, you have all these collaborations going on at the same time to try and get these compounds from the bench to the clinic as, quickly and streamlined as possible.
Right.
So again, is there anybody last call for any last questions? Um, doesn't look like that. Going once, twice. Okay. So yeah, once again, I want to thank Carolina for giving some of her time, as well as giving us such an interesting lecture. And yeah, I guess we'll call it here and, thank you once again. And yeah,
Right.
I'd be ending the call then.
All
Bye.
right. Thank you, Jose. Bye, everyone.